Is okay?
Okay, miss.
Okay, then try to remember some terms in this. Okay,
Okay,
I'm going
miss.
to play it. Yes.
An autotroph versus heterotroph. An autotroph, also called a producer, is an organism that can self-feed. The word autotroph comes from the Greek word autos, meaning self, and troph, meaning nourishing or nutrition. Autotrophs are the producers in a food web, and they are the bottom base in an energy pyramid. Some common examples would include autotrophs produce their energy from the sun in a chemical process called photosynthesis. They take carbon dioxide plus water and sunlight and produce sugar and oxygen.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight.
A heterotroph comes from the Greek word meaning heteros, meaning another or different, and troph, meaning nutrition or nourishing. A heterotroph has to get their energy by eating other plants, animals, or by decomposing. They use sugar plus oxygen to produce carbon dioxide plus water and energy in the form of ATP. This process is called cellular respiration. Some heterotrophs eat only plants, called herbivores. Others eat eat only meat, carnivores.
Okay, Menindu. I'm going to ask you something about the things which given in the video. So when you look at the video, when you are watching the video, uh, what are the things, what are the types of different ways you learn about the nutrition of different organisms? Are there how many ways you have heard from that? Like about animals, how they get their nutrition, how do they take their nutrition or the raw materials to produce their energy, how do they take that? What about your idea about that? Just tell me just few words about that.
by eating foods.
Yes, yes, yeah. By eating foods. Good. By eating foods, they take their nutrition. That is the containing when we eat food, as mentioned, as given here, this lamb uh, drinking milk, and here there's a little girl with some fruits and vegetables with her. So in this way, we are eating food to take our to uh, get our nutritions to produce energy in our bodies. And also there are different ways of nutrition among the animals, we know that. And also watch we saw in that video, some animals eat plants, they are named as herbivores, and some animals eat meat, they are named as carnivores. And animals like us, human, and also dogs and cats, everyone, they are named as omnivores because they eat both food containing meat or vegetable products. So here, that is about animals. And what about the plants? How do they take their nutrition? What is the idea about that? Do they eat food?
No, they are absorbing water from the
Yeah,
underground and
yeah. They absorb water, and sometimes some what are the minerals, those things, fertilizers විදියට අපි දානවා, fertilizers, and sometimes different gases from the atmosphere. In this way they absorb different things, and importantly what they do is they produce food in them, what we can't do the production of food in us, but plant can do. So that special process is the today lesson. So here we can see some plants are absorbing water, and from air they absorb something else, and here you can see there's sunlight, and in that way these plants are growing. That is the way the plants are growing. They grow by themselves, they produce their food by themselves. So that process is here it is mentioned about the nutrition of animals. It is given here. Animals, since they are depending on the foods produced by plants indirectly or directly, we call them as heterotrophs. We saw that in that video. They are named as heterotrophs. They have a heterotrophic mode of nutrition. What is this word? Indirectly or directly? Directly means like this cow, they just depend on plants directly. And these carnivores and some omnivores like us, we depend on plants indirectly sometimes, because these lions, they are eating meat. This meat is mostly it's just made from a deer or a cow or someone else, like any herbivore. It may be eating grass. So this lion is eating that animal. So they are depending on the grass or the plants. Here it is. So printing mistake. And they are depending on the plants indirectly. And here also there are some bivalves and some onions or carrots and like this. We are depending on plants sometimes as a direct way, and then sometimes indirectly. So that is about animals. And then plants, that is about plants. Nutrition of plants. Plants are named as autotrophic organisms because they can produce their own food. That special process is photosynthesis. So today our lesson is to learn about this special process of photosynthesis. So I'm moving to the slides now. This is about photosynthesis. This is just a small illustration about the process of photosynthesis. You can see here there are some essential factors for the photosynthesis. Mainly 4 factors are essential for the photosynthesis to occur. As you told me, water, sorry, water is absorbed from the soil, and then another gas named as carbon dioxide is absorbed from the atmosphere. And then here you can see chlorophyll in leaves. These green leaves contain chlorophyll in their cells. Chlorophyll is a green color pigment which is in green color plant parts. And then they absorb light energy from sun. Those are the 4 essential factors for the photosynthesis. Then this is the definition for the photosynthesis. Here we have to remember this one. Once you are writing the tutorial, you have to answer for the definition of the photosynthesis, and also you have to remember this. Photosynthesis is the process of utilizing light energy, that is the energy source. Like we are using fire or different fuels like LP gas to prepare our meals. That's why we are using fuels or the energy sources to prepare our food. In that way, plants also use an energy source, that is the light energy from sun. So the process of utilizing light energy is a process of using light energy. For what? To produce food in their body, to produce food. For that process, they have some helps for that process, that is chlorophyll in green plants. Chlorophyll is the green color pigment. It has an ability to absorb light energy to carry out this process of producing food. So that is just like the machines in a factory. Assume a plant is just like a factory. It uses electric energy to run the machines. So in that way, if this factory, in the same way that factory is having machines, this plant is having chloroplast, which is containing chlorophyll to utilize light energy to produce food. Like the factory is making particular product by using energy and different machines. Give me a second. So for that process of producing food, they are using carbon dioxide and water. Carbon dioxide gas is absorbed from the atmosphere and water is absorbed from the soil. They are just raw material. As an example, in a factory complex, they are producing particular product. To produce that particular product, they are using different raw material. If it is a bun, they are using wheat flour, sugar, and water, and different raw material, maybe flavors in that way. So for the process of producing food in plants, they use carbon dioxide gas and water. So machines are the chloroplast, which is containing chlorophyll. So that particular cellular organelle which is carrying out this process of photosynthesis is chloroplast. We have to remember that too. It is not mentioned here. Chloroplast. Chloroplast. That is the cellular organelle. It's like this. It has a double membrane in it, and it has some green color plate like structures. Those are interconnected each other. They are just like this. You may have learned this in your grade 10 lesson number 7. Not it's 8. And then we are moving to discuss about the process in detail. Here you can see it absorbs light energy like this. And then again it absorbs carbon dioxide gas plus water. So these are the things get taken into the plant leaf, and then it gives two products. The main products of the photosynthesis is glucose. It is given here, and then it produces oxygen gas. Here you can see it's an image of some chloroplasts in plant cells under microscope. Actually, they are moving like this. So we call this as the cytoplasmic streaming. Cytoplasmic streaming. The chloroplasts in a plant cell are moving around the cell. Here we can see they are moving around the cell, at the periphery of the cell, because they have all plant cells. That means each and every plant cell having a vacuole in it. Since they have a vacuole, they are moving around the vacuole. They are moving at the periphery of the plant cell. So these are some cells of a hydrilla plant leaf. So it's just a very, very smooth and thin leaf. So therefore we can see all the cells in it clearly. So this is the structure of chloroplast, which contain plate-like structures with green color pigment named as chlorophyll. So that is about the structure of the chloroplast and the position of the chloroplast. So that's just for your knowledge. And then here we are going to talk about this main product, which is glucose. In the process of photosynthesis, the glucose is the sugar or the carbohydrate which is produced at first. And then this glucose is converted. It is converted into another type of carbohydrate named as starch. So it is given here. That is starch. And then this starch is going to be stored in a plant for some time, maybe like 2 days, like 48 hours. It is going to be stored in the plant. And then when it is high in amount in the plant, this starch is converted into another sucrose named another carbohydrate named as sucrose. That is sucrose. In grade 10, we have learned about the types of all the carbohydrate. So I'll remind that again. This glucose is a monosaccharide. It is water soluble and it's just a single molecule of monosaccharide. So it is named as a simple sugar also. And then this is converted into starch. It's a polysaccharide and it is stored in the plant for some time, and then it is converted into another product named as sucrose. This sucrose is water soluble. this is transported along a particular specific tissue in our plant's body. So, Minidu, can you remember the type of plant tissue which transport the products of photosynthesis, mainly sucrose? Can you remember, or can you tell me now? What is that tissue? There's a tissue which transport food in plants. Can you remember? No, yes,
Not
what is that? Can't you remember? I couldn't hear you. Did you tell the answer?
no, miss.
Okay. Okay. Now remember. So we were doing the class. We were doing that lesson in the last week, and you were so tired at that day. I continued all the lessons since we are having a small time to complete all the things because now it's already March, and so we have a small time. So I'm doing somewhat faster, but I'll tell you that now. So Minindu, remember to take that paper and answer it. Then you will memorize all of these things and you will remind all of those things again. Okay? Remember to do that, okay?
Okay, miss.
Okay. So the tissue, the plant tissue which transport sucrose or else the products of photosynthesis is phloem. That tissue is phloem. It is transported along the phloem tissue. And we have another tissue which transport water. So remember that is xylem. Xylem is not mentioned here because that is just about absorbing water and it is about this part. But food is transport, the sucrose is transport along the phloem tissue. It's a complex tissue which contain different types of cells. Uh, sorry, cells in that like sieve tube elements and then companion cells and phloem parenchyma and then phloem fibers. Those are the four main types of tissues, four main types of cells in phloem tissue. And then xylem tissue also having that kind of different cells because xylem and phloem are complex tissues in a plant body. So that is just a short memory about the last or the previous lesson. And then now we are going to learn how these things are absorbed, like carbon dioxide, how is it absorbed, and how the water is absorbed, and how the light is absorbed. So we have to learn those things too. I just told only about what is the main product and how is it convert into different things here. And then finally, the sucrose, which is transported along the phloem, is going to be stored somewhere, because plants produce this glucose as their food, not for us. They are not making it for us. It is just for their use, but we are depending on plants. So here, this sucrose is again converted into starch, or maybe into another different types of carbohydrate. If it is going to be, if this sucrose is going to be stored in a yam, or if it is going to be stored in a manioc or something else like a potato tube, in that way it is converted again into starch and stored there. And if it is a fruit, if it is a ripening fruit, it is going to be converted into another simple sugar molecule like fructose, that is named as fruit sugar. Fruit sugar is stored in fruits. So the sucrose is again converting into different things in the plant body. That is the normal way, and that is the normal process of the glucose. What happened to it is that. And then I'm going to tell you how does this water is absorbed. How does the water is going to be absorbed? Water is absorbed from the roots, from the soil mixture. Here, roots are having a special cell that is root hair cells. They are like these root hair cells. When we take a root, you may have seen they mostly when we are soaking some seeds to plant, we can see when they form, when they are forming small white color roots, we can see like woolly structures at the end of these roots. Those are named as root hairs. Those root hairs are increasing the surface area of the root to absorb more water and nutrients. So root hair cell is like this, and other epidermal cells are like this in the root. And on the other side of the root, if we take a cross section, they are also having some root hair cells and other epidermal cells. Then here there are some tissues like xylem and phloem to transport food and water between the roots and the upper parts of the plant. So here, this process of absorbing water into the root hair cells is named as osmosis. That is osmosis. Osmosis. Because soil mixture contain high amount of water, but in root hair cells or inside the root, there's no enough much amount of water. So water coming from the place where there is a high amount of water to the low amount place where there's a low amount of water. So water coming into the root. That process is named as osmosis. That is about the absorption of water. Then this absorbed water travel into the xylem of the root. As I mentioned here, these are the xylem tissues. The xylem tissues take water from the root hair cell. Root hair cell takes water from the soil mixture by osmosis, and that water is sent to the xylem tissues in the root. And then this water is transported along the xylem tissue in the root, and it entered into the xylem tissues in the stem. Stem has again xylem tissues and phloem tissues. Xylem tissue takes the water from xylem tissues of the root. So these are the xylem tissues in stem. They take water from roots. In that way, those are then sent to the plant leaves. Here there's another layer of tissues at, uh, through the from root hair cells to the xylem tissues. In roots, there's another two layers of cells. They are named as cortex and endodermis. When we take a cross section of a plant root, we have the epidermis at first, like this. This is the epidermis, and it contains these root hairs and all the things like this. And beyond that, there's a cell layer named as cortex. This is cortex. What is sent to the xylem through these two cell layers, I couldn't tell you that. This is cortex, 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 this second layer. And then after that, there's a small single cell layer named as endodermis. Endo means ඇතුලෙන් කියන එක. Inside කියන එක. Endodermis. 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 So this outermost layer named as epidermis. So with that you can remember that two words. Dermis means that it's like skin. අර outer area එක හැම skin එක කියන එක වගේ meaning එකක් තමයි dermis කියන එකෙන් දෙන්නේ. ඉතින් epidermis means outer layer. Epidermis. Epidermis is having root hair cells. Epidermis. This is epidermis. Epidermis is having root hair cells, and root hair cells take water in, and then it is sent to the cortex, and then it is sent to the endodermis. And through these two cell layers, actually through these three cell layers, water enters into the xylem tissue in the root. I couldn't tell you about that here, so that's why I drew this image. I'm drawing this image again. So that is the way of entering water. So Minitu, what is the process? What is the name of the special process of absorbing water to the plant from the soil? This name, what is this? What is this special name we are giving to the way of absorbing water, or is the mechanism of absorbing water? What we call that? Osmosis. Can you hear me? Yeah, osmosis. Can you hear me well? If you can't hear me well, you can tell me, okay? Once you have a problem with... Okay. So that is osmosis. So I describe here the way of absorbing water. And then I'm going to discuss the way of absorbing carbon dioxide to the plant. Water is in the soil, but carbon dioxide is in atmosphere. It is absorbing from the atmosphere. So to enter into the plant from atmosphere, plant leaves are having small holes in their structure. When we take a section from these lower sides of a leaf, we can see some holes like this. They are just bean-shaped cells. We call them as guard cells, and in between little pore, this little pore is named as the stomata. So that is the thing which helps the plant to absorb the carbon dioxide gas from the atmosphere. So what is this structure is named as?
Stomata.
Stomata, yes. Stomata is the place where carbon dioxide gas is absorbed. So that special process of absorb or the mechanism of absorbing this carbon dioxide is named as diffusion. Carbon dioxide gas is diffused from atmosphere to the plant leaf. Diffusion. For water, it is osmosis. For carbon dioxide, the mechanism is diffusion. You have to remember those two words and also where they are absorbed from. Water is from root hairs and carbon dioxide gas is absorbed through stomata. So that is the way of absorbing it. And then, as I told you at the beginning, this before that I have to tell you what are the and where it is going to be take in. The place is stomata. Carbon dioxide gas enter into the inside area of the plant leaf through stomata, through this pore, and this is the arrow. And then they are entering into the middle area of the plant leaf. When we take a cross section, they are entering into the middle area. So this area named as mesophyll cells. All the cells in inner area of the plant leaf, they are named as mesophyll cells. So there are different types of mesophyll cells like spongiparenchyma, palisade parenchyma. When we look at a cross section of a plant leaf, can you remember we just look at a photograph of a cross section of a plant leaf when we are discussing about the parenchyma, sclerenchyma, and collenchyma. Can you remember? Here there were some cells which containing chloroplast inside the plant leaf. Those are named as parenchyma cells. Actually, they are parenchyma cells. So they are two types, palisade parenchyma and spongiparenchyma. These both parenchyma cells are inside the leaf, so we call them as mesophyll. Mesophyll at the meso means at the middle කියන එක. Mesophyll cells. So that is the way of absorbing carbon dioxide gas and water. And then I'm going to tell you how the light is absorbed. As I told you at the beginning of the of this slide, but I was discussing about chloroplast. Chlorophyll pigments inside the chloroplast absorb light energy. That is how these three factors are going to be absorbed to the leaf. And then these two things are produced, glucose and oxygen gas. Those are produced. So is that clear, Minindu?
Yes, miss.
Okay. So I am moving to the next slide. So here we learn about the process of photosynthesis. Now we have to know what is the word equation for the photosynthesis. because sorry, because photosynthesis is a chemical process. It's a chemical reaction which is happen in plant leaves. All the metabolic reactions in animal bodies, in plant bodies, they are chemical reactions. So we have a chemical equation for all of these metabolic reactions in our body too. So in the same way, for the photosynthesis, that is a main metabolic reaction in a plant body. So it has a word equation like this. All the raw materials are mentioned in the right hand side of the equation and all the products are mentioned in the left hand side of the equation. Sorry. Uh, two raw materials are carbon dioxide gas and water. They are combined together in the presence of chlorophyll and sunlight. That's why this chlorophyll and sunlight are mentioned with the arrow. That means carbon dioxide gas plus water in the presence of chlorophyll and sunlight, they produce glucose and oxygen. That is the word equation for photosynthesis. So we have to write the chemical equation too. Now this is the balanced chemical equation for the photosynthesis. Here carbon dioxide gas is CO2 and water is H2O, glucose is C6H12O6 and oxygen is O2, and we have balanced the equation by putting same number of each and every element to the both sides. So here we don't have to uh, balance uh, by putting an atom to it. Rather than that, we can just remember without putting anything in front of this glucose formula, we can put number 6 in front of each and every part of this equation. Once we are putting 6 to carbon dioxide, water and oxygen, all the atoms of different elements are equal in number at the both sides. Here six CO2 molecules, 6 H2O molecules combine together to form a glucose molecule and 6 oxygen molecules. Here 6C, carbon higher, and then here another C6. So the carbon is balanced. And now let's look at hydrogen. Hydrogen is 6 into 2. That means 12 hydrogen here. Here we have that 12 hydrogen in this side too, in the products. So it is balanced. Here it's 6 carbon, here it is 6 carbon. And what about the oxygen here? Here there is 6 into 2, 12 oxygen atoms in this CO2 molecules. And then there are 6 water molecules. They are having another 6 oxygen atoms. So sum of the all oxygen atom is 18. Now let's see whether there is 18 oxygen atoms in this side. So 2. Here it is 6. 6 into 2 it's 12. So 6 plus 12, we have another 18 oxygen atoms in this side. So in this side too. Can you see, Minindo? Is that clear?
Yes, miss.
Yes. That is the way of balancing it. So no need to try hard to balance this by putting all the numbers and like this. Just you can remember, put number 6 to CO2, to H2O and to O2. Without putting any number to glucose, you can balance the chemical equation of the photosynthesis. Okay, that is the equations. And then I am moving to tell you about the products and raw materials. Oh my God, it has changed here. Give me a second. Those are the products, those are the raw materials. I have put that wrong. Okay, here. These are the carbon dioxide and water are the raw materials. We were discussing that from the beginning too. They are raw materials. And products are the glucose and oxygen. So here, glucose is the main product. Oh, glucose is the main product of photosynthesis. It is the main product. We have to remember that. That is main product. That is main product. And oxygen is just a byproduct. It is just a byproduct, but it is really valuable for us because we are respirating oxygen gas. It's a byproduct. It is not required for the plants to, to a high amount. They are also respirating. That's true. They need oxygen. But it is very valuable for us because we can't respirate. We can take any other gas inside our respiratory system rather than this oxygen. We need oxygen for the respiration. So glucose is the main product. Oxygen is the byproduct. Remember that I have put what are the raw materials and what are the products here. And then we are moving to discuss about some test. Here we until now we have discussed what are the basic things about photosynthesis. That means what is photosynthesis and what is the process of photosynthesis and how the different raw materials are absorbed and how the different products are formed and what happened to them at the end. I was discussing what happened to the glucose and I was discussing how the water is absorbed, how the carbon dioxide gas is absorbed. Now we have learned well about the mechanism or the process of photosynthesis. Now we are moving to test different factors whether they require or not for the photosynthesis. And also we are going to test whether the glucose is actually producing in the plants at the end of the photosynthesis. So these are some tests, those which have designed for the uh, confirmation of the production of glucose. Finally, it is stored as the starch in leaves, but that is the product of photosynthesis. We are going to test that, and later on we are going to test uh, is the plant actually absorbing carbon dioxide for the photosynthesis, and um actually water, not the water, light energy or the chlorophyll is required or not for the photosynthesis. We are going to test those things. So at first I am moving here to discuss about whether starch is produced or not at the end of the photosynthesis. Here this is the test. Here you can see there's a beaker. It contains some water and we have put a green leaf into it. And then we are boiling that water sample. Now, our green leaf is in a boiling water sample. We have a beaker and boiling water is in. We have put the green leaf. Green leaf is boiled to some extent. And then what we do is, this is the first step, leaf is boiling in boiling water. And then we take that leaf out of boiling water and put it into a test tube containing alcohol or ethanol. We are putting it into a ethanol test tube. Ethanol is an alcohol. There are different types of alcohol molecules, alcohol compounds, but here we have taken ethanol. Then we are putting this leaf here and again we are going to boil it. What we do actually here is we take the leaf out of this boiling water sample and put it into a test tube containing alcohol and put that test tube again into that boiling water beaker. So now we call it as water bath. We are putting ethanol containing tube in a water bath. Now it is boiling in this way for some time. until the leaf become pale yellow in color. At the beginning, it is green color. You can see normally green leaf is taking for this, and until it become pale yellow color like this, we are going to boil it. And in this way, we can see that alcohol sample or the ethanol sample, it is getting green in color because all the green color pigments in the plant leaf has coming out. They have come out. That's why leaf become decolorized. All the chlorophyll pigment has come out because they are dissolving in ethanol. Chlorophyll is dissolving in ethanol. Leaf doesn't have chlorophyll. And then we take that decolorized leaf or the colorless or the pale yellow color leaf out of the ethanol sample and we put it into a cold water sample. Then we are washing it to remove if there is any uh, chlorophyll pigment on that. We are just watching it and then we take it out. We put it on a white porcelain white porcelain tile or somewhere else or on a paper. We are going to put it somewhere and now we are adding iodine drops. Few drops of iodine is added to the leaf. So in grade 10, lesson 1, you have learned that starch test. To test whether there is starch in our particular food sample or something given to us, what we have to do is we have to add some drops of iodine. So it will give a nice color change from a yellow brown color to blue black color. Bluish purple color එකක් අපිට හම්බෙනවා. That yellow brown color is changing into that blue color only in the presence of starch. So mostly when we take a green leaf from a tree where there is in uh, under sunlight, we just pluck the leaf and do this test. So all the leaves in the environment are photosynthesizing. So they have stored some amount of starch in each and every cell. So we will get this color change at the end. So that is the process of testing whether starch is produced or not at the end of photosynthesis. Mendu, did you understand that?
Yes, miss.
Yes. Okay, that is the process of this test. That is the way of doing this test. So at the end of this test, we have an observation. Our observation is pale yellow color leaf turns into blue-black color when we are adding some iodine. That is our observation. And we can make a conclusion with that starch is produced at the end of the photosynthesis. That is our conclusion. We have concluded that starch is produced in photosynthesis. That is our conclusion. You have to remember that too. And here, when we are given this diagram in our question paper, we are getting some extra questions like they ask us why this ethanol containing test tube is put in a water bath. They have to boil it in the direct flame. Why didn't they do that? We have to answer. The reason is ethanol is flammable. When we hold this ethanol containing tube in a direct flame, it will make a fire. It will get burned at once. So to prevent that kind of accident, we are putting it in a water bath. And sometimes they will ask why the colorless ethanol is turned into green in color at the end. So we have to tell all the chlorophyll pigment in the plant leaf has dissolved in ethanol sample. That is the reason. Chlorophyll in leaf is dissolved in ethanol. That is our answer. So that is our test here. So they are given here. Here there's another thing. Plant leaves are boiled in water to make the leaf porous. That's why they are boiling this green leaf in a water sample. Actually, when we look at this, it's like there's nothing with that. But actually, there's a reason for that. The leaf become little bit porous. Hot water is making some little pores, so it is easy for the chlorophyll pigments to release out when it is put in the ethanol mixture. Otherwise, if we put this green leaf directly in the ethanol mixture without going with this step, we won't get the uh, uh, like we won't get the most correct results, because this green color is not disappearing fast. We have to boil it for a long time until the ethanol is evaporating. We will have to boil that, so it won't work. That's why we put it in boiling water and make it porous and make the process of removing chlorophyll easier. That's why we do that. And then plant leaves are boiled in an alcohol solution to remove chlorophyll pigment. We know that. An alcohol containing test tube is boiled in a water bath without putting it in a direct flame, as alcohol is highly flammable. Because to prevent an accident, we are doing this. And then that is about the main product of the photosynthesis. So now we are going to discuss about the um, about some tests for the factors required for the photosynthesis. Minindo, can you tell me, uh, can you remember what are the 4 factors required for the photosynthesis? Up to now, we discussed it at the beginning.
Chlorophyll, light energy, water, carbon dioxide.
Yeah, good. Those are the main 4 factors required for the photosynthesis. So we are going to discuss here whether these factors are really required or not. But we are not going to test about water due to a particular reason. I will tell it at the end. But here we can test light energy, carbon dioxide, and chlorophyll. So this is about light energy. Here what we have to do is, we have to take a potted plant, a plant which was planted in a pot. We have to take it, because we have to keep it under dark conditions for 2 days. That means 48 hours. We are putting it in inside a cupboard or somewhere. If not, we are going to put a black color or a tulip bag and then black paper, whatever we are using to cover it for 2 days. So select 2 plant leaves from a potted plant which was kept in dark for 48 hours. That is our first step for the test. We have to take that plant which was kept in dark for 48 hours, and we have to select 2 similar leaves. Here they have selected leaf A and B. Those are similar leaves. They have covered the leaf A with a black strip of paper or black color strip of polythene. And then not the whole leaf, but just a strip of that. Just a part of that leaf is covered like this with a black color polythene. We cover it and put some clips like this. That is the way they are normally doing this. Cover it, clip it. And then we have to take another colorless polythene paper and put it in the same way as this black polythene. So black polythene is our test. This is our control. Colorless polythene is our control. Black polythene is our test. So normally when we are doing a test, scientific experiment, we are doing the test with the present of a with the present of a control. Control is having same conditions like the test, but without the factor we are going to test. So here this A, leaf A doesn't get sunlight due to the presence of this black polythene, but colorless polythene is there. It gets sunlight, and they are also taking water in the same way, and they had chlorophyll at the beginning in the same way because two similar leaves, and also um, they are having all the nutrition, same nutrition and all the same things. That's why we take the control like this. And then as the second step we covered. And then as the third step, keep the plant under sunlight for 3 to 5 hours. We have to keep this set up under direct sunlight for 3 to 5 hours. 3 to 5 hours තියන්න. Then we have to take that plant and we have to detach these two leaves. Then we have to do the starch test, which we have performed here. We have to do this starch test for the plucked two leaves. We take those two leaves plucked and put it in water bath to boil like this, and then these two leaves are kept in ethanol and boiled again, and it is decolorized and become pale yellow color. In our lesson we use this word pale yellow or white. We can use any. It's just like mostly it is pale yellow in color. And then we have to do this starch test like that. So as the observation, we will get something like this. In leaf A, which was covered with the black polythene paper, it has a blue color area in both sides of the leaf except the area covered with the black polythene. There's no color change when we put some iodine on the pale yellow color boiled leaf. This is pale yellow color. It is pale yellow color. This area is a blue color. And here in leaf B, which was covered with a colorless polythene, it is becoming dark purple or blue color. Bluish black, different words are using for this. Bluish black, dark purple, dark blue, whatever. So that is the color change which is given by iodine in the presence of starch. So observation, covered area of leaf B turns into blue-black color, but the covered area of leaf A didn't give a color change. So our conclusion is, oh, it's a printing mistake, so I have to correct it here. Light energy. Light energy is essential for photosynthesis. That is our conclusion. We know that light energy is required to produce starch. Producing starch is the photosynthesis. So light energy is required for the photosynthesis. No photosynthesis here, but photosynthesis has happened in this leaf B due to the presence of sunlight. And then, Vinidu, did you understand how it become blue-black color like this? And how this area doesn't become blue-black color? Did you get it?
Yes, miss.
Okay. Is there any problem? You can ask if there is some.
No, Miss,
No?
I'm
Oh,
clear.
yes. Okay. Okay, I'm moving to the here we have to answer a question. Why should we keep the plant in dark for 48 hours? There's a reason for that. I told you that, uh, sorry, when we were discussing about the process of photosynthesis, I told you that glucose is produced at first and then it is converted into starch and stored in the plant leaves for some time. And then finally it is converted into sucrose and transported to the places where it is going to be stored. So this is the reason for keeping the plant in dark for 48 hours. Normally, the glucose which was converted into starch is going to be there for about 2 days, 48 hours. 48 hours. Once we are keeping this plant under dark for 48 hours, all the starch previously produced and stored in the leaf, they are going to be converted little by little into sucrose and sending away from the plant leaf. Then, since it is in dark, it is not doing the photosynthesis now. So, new glucose is not producing to convert into starch. And the produced starch also has converted into starch and sent out. Now, this plant leaf doesn't contain any starch. That's why we removed this starch at the beginning because we want to test whether starch is produced or not during the photosynthesis. But if we have photosynthesis at first, we won't get these two results. We will get two purple color leaves like this. But we have removed all at first and then we are keeping the plant in sunlight, under sunlight for 3 to 5 hours. That's how we did that. So we did that. The purpose of doing that is to remove starch completely. To remove all the starch completely is the reason for keeping it in dark. Then I am moving to next. That is about carbon dioxide gas. To test the carbon dioxide is required or not, we have to use this setup. Here we have a potted plant again, as in the same way in the previous test. We have a potted plant which has kept under dark conditions for 48 hours. Again 48 hours we have to keep it in dark. And then we take it out, we select two leaves. We are not putting it under sunlight while we are doing this setup because it will do photosynthesis again. So our test is again going to be wrong. So we keep it there and select two plant leaves named as C and as labeled here as C and D. Then we take another two polythene bags. We put some water into one and put some KOH solution into uh, other polythene bag. So this KOH has a special ability to absorb CO2. That's why we are putting it there. It absorbs CO2. It absorbs carbon dioxide. Then we insert those two leaves separately into these two polythene bags containing water and KOH solution and make them airtight. We have to put a rubber band or something to prevent the flow of air in and out. We are tying it tightly. And then we are going to keep this as in the previous test in sunlight. We are keeping it under sunlight for 3 to 5 hours again. So say same procedure, but two different uh, chemicals or setup. So then we again detach the leaves as previous test, as done in the previous test, and we are performing the starch test. So our result is this. In D, no color change. D was kept in a cavoid solution containing polythene bag. And C, there's no problem with C, no KOH to absorb anything here, to change anything in it. It has water, only water, and it carrying out the photosynthesis well. So our result is dark purple or blue color leaf at the end when we are putting iodine. And in case of D, there's no color change because in that airtight polythene bag, all the carbon dioxide gas present in is absorbed by this KOH. All the carbon dioxide is absorbed by KOH. Then there's no carbon dioxide in it to absorb by the plant. Carbon dioxide cannot be taken in from the outside too because it is airtight. Then there's no carbon dioxide for it, there's no photosynthesis, there's no starch in it to give a color change with iodine solution. So that is it. So our observation is the leaf C become blue black color and leaf D didn't give a color change. So our conclusion is carbon dioxide gas is necessary for the photosynthesis. Here it is mentioned that we get these results because the KOH in um, D leaf with leaf D absorb carbon dioxide gas. So that is about the test for the checking whether carbon dioxide gas is essential or not for the photosynthesis, and that is our results. Now I am moving to the third factor we are going to discuss about. It is chlorophyll. So here chlorophyll is required for the photosynthesis. We know that, but we are going to check it here. So what we have to do is we have to just pluck a leaf from a plant like this. We have seen this kind of croton plants with green and white color patches. Green color patches and white color patches are there. We have to select a leaf like that. So we can select this leaf here because we are selecting, I select this leaf is, here in this test we have to draw a sketch of this uh, colored mosaic pattern. If we take a leaf like this, we will get into a trouble. We can't draw all the things like this, and we can just mark the areas, but this is somewhat clear. We have few patches of yellow color area and other area is green in color. So now I have marked all the white patches in that leaf. We are just drawing this in a paper. And then what we do is we carry out the starch test. Starch test is performed and this is the final result. Before the starch test, we have a leaf like this. We have a leaf like this with green color leaf with some white color or yellow color patches without chlorophyll. And then later on, it turns into a pattern like this. All our observation is all the green color area turns into blue black color or the dark purple color, but the white patches or the yellow color patches didn't change into blue black color. The reason is chlorophyll is necessary for the photosynthesis. Actually, that is the reason, and also that is our conclusion. Chlorophyll is an essential factor for the photosynthesis. is an essential factor. We have to use that word, essential factor. Essential factor is an essential factor for the photosynthesis. And then our fourth factor, it is water. Normally, this is not going to be tested in the normal laboratory conditions like a school laboratory, because once we are going to keep a plant as a test without giving water to it, and we are giving water to one plant, we are not giving water to another plant, which is used as the test. So control it is going to be grow and be alive due to the presence of water, but our test going to be die. It's going to be wilt and die uh, due to the absence of water. So we can't do this in a laboratory, but scientists have performed a particular method. They have used a particular method to check whether water is absorbed or not from the plant. What they have done is they have used water labeled with oxygen 8 18 isotope. Oxygen 818 isotope is not a normal oxygen atom. Normal oxygen atom, we know that it is oxygen 816. That is the normal oxygen we know, 816. But this is somewhat different. This is a radioactive oxygen atom. And once it is used to produce some water molecule, that scientists have given that water to a particular plant. And finally, they have collected the oxygen gas removed from that particular plant, and they have checked whether that oxygen gas contain that oxygen isotope or not. So they have observed that oxygen 818 radioactive isotope is present in oxygen gas. So they have told that water is essential for the photosynthesis. We can't do this in a normal laboratory. So that is all about the checking or the testing the testing whether these four factors are required or not for the photosynthesis. Now I am going to discuss about another practical which is in this lesson to discuss. Here now we have done all the things like carbon dioxide gas. When we think about this equation, here we have discussed about carbon dioxide gas. That test we have done with KOH and water. We cannot do this. It has done by scientists by using a radioactive isotope. And chlorophyll, we test it with a mosaic leaf. And sunlight also test in the same way like carbon dioxide gas with a potent plant. And then we have checked whether glucose is produced or not. Not the glucose, but we have done it for the starch. Now we are going to discuss about the final one, oxygen. So this is that setup. Here what we have to do is we have to take some pondweed. Pondweed means maybe hydrilla, vallisneria, whatever the aquatic plant we have to take. And we have to keep that hydrilla, or here it is mentioned that it is as an aquatic plant. We keep that aquatic plant in a funnel, glass funnel we are using for this. We take a glass funnel and put that hydrilla in it, vallisneria in it, and we take another beaker which is filled with water. And then what we do is we keep that funnel containing aquatic plant in it. We uh, put it upside down to the beaker, and then we filled another boiling tube or test tube. Mainly it is easy if we can use a test tube, but boiling tube is okay. We take a boiling tube and filled it with water, and then without taking it out from the beaker, or maybe instead of beaker we can use a basin, water basin for this. We can uh, fill that boiling tube with water and put it upside down on the neck of the funnel inside the water without taking it out. If we taking it out, it will make a gas bubble at the beginning here. So it's not good. It's not the correct method. Here, what we want is at the end of the test, we have to keep some gases in this. But at the beginning, if we keep something like this, it's wrong. At the beginning, this should be filled with water. So this is the setup, and we prepare it and put it under sunlight. After some time, we can see that small air bubbles are releasing from the neck of the funnel, and then they are going to be collected inside the at the end of the boiling tube or test tube. Then they collected there. Then once it is having just 2/3 of the test tube, which is uh, containing the air in it, we take the test tube out without removing this, and we have to put our thumb into the opening of the test tube and take it out. Otherwise that is going to be released out. That air bubble is going to be released out, so we have to be careful not to release that out. Then we take that and we test it for a we test it with a particular thing to check whether it is oxygen or not. That is what we do here. So that is the normal setup, and this is a actual setup of that. And you can see here there's an area with containing which containing some gas. Here also you can see in this upper area there's a collection of some gas. We don't know what is this gas is actually. Since we have learned about the photosynthesis, we know it should be the oxygen gas, but we are going to test whether it is oxygen or not. So these are the steps of that practical. Add water into a trough, place some hydrilla or vallisneria plants inside the funnel, fill a boiling tube with water and place it inverted on the funnel inside water. Place the setup in sunlight and let the gas bubbles to release from the aquatic plant and get collected at the top of the boiling tube. Finally, this is the final step. Insert a glowing splint to the test tube. That is done to check whether this is oxygen gas or not. So we have to take a glowing splint. Most probably it will be, we can take a dry eagle, or we can take an incense stick. Mostly we are taking an incense stick or a dry eagle. Then we take that and burn it. After burning it, we can see a glowing end at the end of that. So we take that and insert into our test tube without removing that air bubble in it. We have removed water out. Then we are entering our glowing splint into it. Once we enter it, we can see this kind of observation. Initially, it's a glowing splint without a flame, but then we can see a nice bright flame is forming due to the presence of oxygen gas, because oxygen is a uh, burning friendly gas. That means it is the gas required for the burning or the combustion. That's why we use this glowing splint to identify the presence of the oxygen gas in the releasing gas from the photosynthesis. So that is our test. So we have come to the end of the lesson. Now we have done the process of photosynthesis and some tests to identify different factors required for the photosynthesis and test for the two different products of the photosynthesis. And finally, I'm going to tell you what are the importances of this photosynthesis. So, as I told you at the beginning, it provides all the food directly or indirectly for all of the animals on this earth. And therefore, it is essential for the maintenance of the life on earth. Then, uh, I told you that as a byproduct of the photosynthesis, oxygen gas is produced. So, it is the source of respiration of each, all of us. As aerobic organisms, we require oxygen for the respiration. So, it is given by uh, photosynthesis. And then again, oxygen is required for the combustion. We can't burn anything without oxygen. That's why in our small grades, we are using this kind of a practical. We take a glass like this, and initially what we do is, sorry, initially we take a candle and light that and put it and paste it on a table and place it somewhere and close that candle with a glass. Then we will observe that this candle is going to be, the flame is going to be lower and lower, and finally it is removed. So it is blowed out. So the reason is oxygen gas is required for the combustion. Then the third importance of the photosynthesis is it absorbs all the carbon dioxide gas and maintains the oxygen carbon dioxide balance in the atmosphere. It absorbs carbon dioxide gas to produce it food. The plant absorbs to produce food. But actually, carbon dioxide gas is a critical factor which control the atmospheric temperature. In nowadays, due to the high emission of vehicle, due to the high amount of vehicle emissions and also the smokes coming out from the factories, so due to those things, carbon dioxide concentration in this atmosphere is very high, and also we are cutting down all the trees one by one. So carbon dioxide absorption from the plants also getting lower, emission getting higher, and absorption getting lower. So at the end, the amount of carbon dioxide in atmosphere increase. So oxygen carbon dioxide balance going to be, it's going to be unbalanced. So we will get the global warming, and we will get a numerous amount of environmental problems like increase of the sea level and dissolving of the glaciers, and all the things happen. So this maintaining this balance is done by plants. So it is really important for this environment. And then it helps to maintain the carbon cycle. It absorbs carbon dioxide and it produces food. This food is eaten by all the herbivores and omnivores like us, and we eat those things. And then finally, this animal, give me a second. Okay, Minidu. Okay, so it helps to maintain the carbon cycle. There was a disturbance here, that's why I switch off the camera and stop. So it helps to maintain the carbon cycle. As I told you, it is the way of um, like it absorb carbon dioxide and produce food. So it produce food for us. And when all this organic matter going to be decay, all the plants and all the animals and dead organic matter, like dead bodies of animals and plants, when they all going to be decay, they are going to be deposit on this ground. Finally, they were turning into fossil fuels. So that is the overall idea of a carbon cycle. Then fossil fuels are burned by the human, and then it is going to be again released to the atmosphere. In this way, carbon molecule, sorry, carbon atom is going to be cycled all over in this atmosphere. Carbon is going to be cycled in the carbon cycle. This photosynthesis is the main mechanism which helps for this process. So that is the overall importance of the photosynthesis. So Minidu, at the end, do you have any problem with these things I told you? And do you have to ask any questions?
No, miss, I'm clear with it.
I clear it. Okay. So shall I ask questions from you then? Is that okay? Shall I? Some questions?
Miss, sorry, I didn't hear you.
Okay. I was asking that. Shall I ask you some questions then? Is that okay? I'm going to ask you few questions. I was asking questions from you. So here I'm going to ask you that. What is the chemical compound which was used by us to absorb carbon dioxide gas in a test to check whether carbon dioxide gas is essential for the photosynthesis or not? Can you remember the name of that chemical?
Miss, I can't hear you properly.
Uh, I was asking
I
you.
think I have some internet issues.
Yeah, that's true. Okay, Minidu. Then I'm going to tell it like this. uh, I was asking you, what is the chemical compound we used to absorb carbon dioxide gas when we are testing whether carbon dioxide is essential or not for the photosynthesis? Here, what is this chemical? Can you see this? This is KOH solution, potassium hydroxide. You have to remember this kind of little things like this. Uh, you will it won't be a problem when you take a printout of the tutorial and fill it. Okay, Minindu. So I am going to finish the class today. So you have to download 2 tutorials and 1 paper and fill the answers and send me the photographs. Mainly, I want the photographs of the paper. Okay.
Okay, miss.
Okay. You can take some time and fill. You can send it back. Actually, you can upload the photographs back to the website. So, Minindo, I'm going to stop up to now. So, good night, Minindo.
Miss, sorry, I didn't hear the last thing you said. I have some internet issues.
Yeah, that's true. I was telling you to take the printouts later and answer the questions on those things and prepare your notes correctly with tutorials. You have tutorials and you don't have to write a note in a notebook. You can attach those tutorials together and prepare a complete note. Could you hear that, Minindo?
Yes, miss.
Yes. Okay, so we are going to stop up to today. So good night, Minindo.
Good night, miss. Thank you.
You are welcome.